As a doctor in training, I remember admitting a patient named Destiny while on call to the hospital. Her mother brought her in. She was limp and moaning, and her eyes seemed really big on her very pale face. Her mother said her knee was in pain and she was limping. We did blood tests, and the results showed what we feared. She had leukemia, the cancer of the blood. Leukemia starts in the bone marrow where blood starts. As the leukemia grows, it actually pushes the bone from the inside out. That's what causes the knee pain and the limping. The leukemia also crowds out the cells that create red blood cells. That's what causes the anemia. That's why her face is pale, and that's why her eyes look really big, because the red vessels in her eyes disappear, causing really white eyes. Unfortunately, this is something that happens around the world 14 million times a year. I never found out what happened with Destiny since she was transferred to another hospital. Undoubtedly, she received 3 years of chemotherapy and maybe even some radiation therapy. I hope she's a happy, healthy teenager right now. Destiny's diagnosis, childhood ALL, acute lymphoblastic leukemia, is a cancer that has a pretty good outcome with current therapies. But some cancers do not. That's why I'm so excited about Bexion and their drug BXQ-350, because it promises to treat all cancers. It was discovered by a researcher working on a very rare disease called Gaucher disease, which is unrelated to cancer. During my 4 years of medical school, I faintly remember one part of one lecture that covered Gaucher disease and the other lysosomal storage diseases. Even in my pediatric textbook, which you can see is very thick and very detailed, there's only one part of one page that talks about Gaucher disease, which is right here. This is understandable since Gaucher's disease is a 1 in a million disease. About 7,000 to 10,000 people are afflicted with the disease worldwide. What I hope to cover with this video is, how does a researcher who works on Gaucher find a therapy for cancer? In fact, the 3 questions I want to cover is, what does it look like in real life? What is it? What is the drug made out of? How does it, Number 2, how does it work? And number 3, why does it work? Or a general structure, framework, how it works. Now, I'm not an expert or a researcher in Gaucher disease, and I'm not an oncologist. However, as a doctor, I think there are a few things I can provide. 1, I don't come with biases from the world of human genetics or cancer. Number 2, I hope to find a connection between these 2 diseases. And number 3, since I'm not an expert, I hope I can detail this in language that's understandable to all doctors and patients and others. It is often said that the biggest breakthroughs in medicine come from the most unexpected areas, and lots of other fields also. That innovation comes when you take one idea from one area and apply it to another. That's called outside-the-box thinking, and that's what BXQ-350's story is all about. What we have here is a schematic, a chart, if you will, that details what outside-the-box thinking looks like. This chart represents inside-the-box thinking, current therapies today, such as chemotherapy, radiation therapy, anti-angiogenesis, personalized precision medicine. This bigger box represents all cancer knowledge, and the biggest box represents all knowledge of medicine. These 3 little boxes represent outside-the-box thinking. Your first box is information about medicine that does not correlate with anything in cancer. We'll be covering basically Gaucher disease. The second box talks about cancer research, but not related with the current therapies. And this third box takes those 2 ideas, connects them, and that details how BXQ-350 attacks cancer. This first box I've entitled The World's Greatest Laboratory. We'll be covering a few areas in this box. First, we'll be doing a perspective of the history of science. Second, we'll see why BXQ-350 comes from the world's greatest laboratory. And third, we'll show how BXQ-350 was born from Gaucher disease. This guy became king by 12 years of age. Over the course of his life, he conquered 6 more countries that surrounded his, and he upgraded his title from king to emperor, and he became Emperor Qin, the first emperor of China. After conquering the known world, he wanted to conquer death itself. He wanted to become immortal. The doctors and scientists at the time felt that mercury, that liquidy metal substance, must have special life-giving properties. Of course, they were fabulously wrong, and he died early. Of course, people in many countries and many cultures figured out that living things work a lot better, like plants, the leaves, and the roots. Unfortunately, plants have multiple ingredients, which can give you multiple effects. But over the centuries, people have been doing trial and error to improve this process. The next big breakthrough with drug development was the birth of organic chemistry, when urea was synthesized from inorganic materials in 1828. Perhaps you've seen some classmates in college carry this around, or you used it yourself. This represents the building blocks of organic chemistry: carbon, hydrogen, oxygen, and nitrogen. These 4 ingredients, these 4 elements, represent 96% of our body. Calcium and phosphate, our bones, represent another 3%. During the years, organic chemistry has eventually led to a field called medicinal chemistry. And this field develops drugs. It isolates and purified the drugs that came from plants, getting single agents, and also created brand new drugs from chemical structures. The great majority of drugs are based on organic chemistry. If you look at the structures of drugs anywhere, most of them look like this. They're made out of oxygen, hydrogen, nitrogen, and carbon. And it's better tolerated by the body that has oxygen, hydrogen, nitrogen, and carbon. Also, structure determines function. With organic chemistry-based drugs, they are small, which is one feature of structure. They go into the blood vessels, bloodstream, they diffuse out, and then they diffuse into cells. As it diffuses into cells, its structure, the small structure, will match a particular protein or enzyme and will inhibit it and cause a particular clinical effect. Unfortunately, these small organic chemistry-based drugs can also diffuse and enter other cells, maybe in even a different organ, block different kind of proteins and enzymes, and cause unintended effects, what we call side effects. Medicinal chemists are now working in integrated teams with structural biologists and computer experts in the hopes to develop a more rational drug system. If the first big breakthrough was knowing to use plants versus inorganic materials, and the second big breakthrough was organic chemistry and making small organic chemistry-based drugs, then the third big breakthrough was the discovery of biologic materials, biologic drugs. The first one discovered in 1920, though it probably was not called or biologic at the time, was insulin. Now, insulin and other hormones like glucocorticosteroids and thyroid are big molecules, and the largeness of the structure inhibits it from entering cells, diffusing cells, unlike small organic chemistry-based drugs. Structure determines function. However, its structure does match perfectly with particular receptor proteins on the surface of cells. And once that happens, a signal occurs on the inside of a cell that produces an effect. Also, since we know that these already exist in the body, that they're very well tolerated. A couple more examples of biologic drugs are erythropoietin and Neupogen. The type of structure that we most associate with biologics are antibodies. Antibodies have 2 structures. One is the constant part, which interacts with the immune system. And the second part is the variable part, which attaches to its target. Antibodies, just like hormones, are too big to enter cells by themselves. And the side effects of antibodies are usually fever, chills, nausea, and effects of wherever the target happens to be. So, looking over the course of the history of science and drug development, we can look at a couple trends. And they relate to how closely the drug fits or what's inside the body. For example, mercury has very large side effects and can cause even death. Plants are at least made out of living material, which is what our body is. Organic chemistry has the 4 ingredients: carbon, hydrogen, oxygen, nitrogen, which our body has. And biologics are large molecules that actually exist in our body, so they're very well tolerated. And in terms of causing a desired effect, again, the biologics are the best because they correspond with what's in the body, and then organic chemistry, small molecules, then plants and mercury. You could argue that biologics are the best drugs that we have, and that because they come from the body, that the body is the world's best laboratory, the world's best drug development system. Now on to BXQ-350. Let's answer a couple of those questions. What is it? What does it look like? How does it work? We know that BXQ-350 has 2 components that are from the human body. They're biologics. So we already know that it comes from the world's greatest laboratory, the world's greatest drug development system. And we know, since they're biological entities, that the side effects are probably very less likely, since they should be well tolerated. As you see here, BXQ-350 has 2 components. One is phosphatidylserine, and one is sapacin C. They are combined, and they can form a monolayer of a nanovesicle, averaging in 190 nanometer diameter. They're too large, like the other biologic molecules, to enter via their size alone. The first component is phosphatidylserine. Phosphatidylserine is part of a family of biologic molecules called phospholipids. There are a few others. For example, there's phosphatidylinositol, phosphatidylcholine, and phosphatidylethanolamine. As you see here, the phospholipids help form membranes on the outside of the cell with 2 membranes, and they also help inside cells, the organelles, which also have membranes. As you can see, there are a couple differences with the phospholipids. For example, phosphatidylcholine is neutral, has no charge, and the cells actually exert energy to put phosphatidylcholine on the outside. And that makes sense because phosphatidylcholine is neutral, and the outside of cells are pH 7 neutral. Phosphatidylserine have a negative charge, and usually they're on the inner layer, the inside layer of the cell. And they're also noted to be on the inside layer of the membrane of a lysosome. This makes sense because the phosphatidylserine's negative charge can help buffer against the acid, which has a positive charge. The second component of BXQ-350 is sapacin C, a protein. We know from the structure that it has 3 functions. First, when acid is added to sapacin C, it can become activated. Second, sapacin C can destabilize membranes and actually cause fusion of this membrane with other membranes as long as there's phosphatidylserine that it can attach to. And 3, inside the lysosome, sapacin C, when combined with phosphatidylserine and activated by the acid, can connect with an enzyme and drive different lysosomal enzymes to produce their components. The purpose and what sapacin C does every day is to recycle glycosphingolipids, a kind of lipid that we have that's different from the phospholipids that we discussed before. Phospholipids are kind of like an ocean on the membranes of cells, and glycosphingolipids are lipids that float on top of it. When they aggregate, they form what we call lipid raft. Lipid rafts are known to help with cell-to-cell communication or help proteins connect with each other on the same cell. When glycosphingolipids need to be recycled, this is what happens. They start on the outside of the cell, they get a little inward blubbing occurs, it passes through an endosome, a kind of organelle, and then eventually the endosome connects with the lysosome. Now when the lysosome connects with the endosome, it's the sapacin C that's helping the process. The acid activates sapacin C. The sapacin C, as we noted before, will destabilize the membrane and then connect with the endosome. And then once the glycosphingolipid, the GSL, connects with the sapacin C and the lysosomal enzyme, it gets split into different parts. Sapacin C activates a number of lysosomal enzymes. A few of them are glucosylceramidase, acid sphingomyelinase, and galactosylceramidase. The only people who study sapacin C and glucosylceramidase are people who research Gaucher disease. There are 3 types of Gaucher disease. We're going to be talking about type 1. With type 1, there's a gene defect that decreases the enzyme that's in the lysosome. And let's look at this reaction. When there's a decrease in this enzyme, the glucosylceramidase, this enzyme inside the lysosome, it stops and inhibits this area, and that causes the substrate, the glucosylceramide, to increase to very high levels. This is basically a lipid of fat, and it grows in high levels in lots of cells, but mostly in the white blood cells and red blood cells. Now the red blood cells with type 1 Gaucher disease get really, really fat because of all the buildup of the substrate. When red blood cells become unstable or old, the body gets rid of it. The way the body gets rid of it is with a cell called a macrophage. Now here's a red blood cell with lots and lots of fat, and the macrophage has acid which engulfs and acidifies and breaks down most red blood cells. Unfortunately, with Gaucher disease type 1, there's so much fat that gets absorbed by the macrophage becomes big and fatty, and it's basically a fatty macrophage, and we call those Gaucher cells. Gaucher cells are so big and fatty that they end up getting stuck in different organs in the body, such as the lung, the bone marrow, and the spleen. Here's a spleen, and the spleen usually sits underneath the rib cage and is protected by it. When you visit your doctor and you're laying down on the exam table, and if he or she asks you to take a deep breath, they push down your abdomen and push up. And they try and feel your spleen. If they're able to feel the spleen, your spleen is too big, from, for example, mononucleosis. And if it's too big, it's at risk for getting fractured, a splenic fracture, which can lead to massive internal bleeding. As you can see here, the spleen is very red, and it acts like a filter to filter out the old and defective red blood cells. And it looks something like this. So if your spleen's a filter and there's a blood, it'll filter like this. Now with Gaucher disease, since you have so many fatty macrophages, this is what happens with the spleen. As you can see, with Gaucher disease, all the fatty macrophages will build up, and then it'll look something like this. You get a big, large spleen that's very fatty, and they tend to be really big and push down into the abdomen. That was the basics of Gaucher disease, but what we're going to do next is a bit more esoteric. We're going to be covering a very rare occurrence with this very rare disease, and this might be the crux, the turning point for this presentation. So with Gaucher disease, you get a lot of the Gaucher cells that are very fat and get stuck in the spleen, as we've talked about before. But there's a second kind of cell that's researchers and scientists have noticed in the spleen that are apoptotic, cells that have undergone programmed cell death. Now these cells, the cells that have died, are not from Gaucher cells that have suddenly died. It came from a separate process. Now if we revisit our formula that we talked about before, with the most common type of cell, you get the blockage of the enzyme, you get very high levels of the substrate, the glucosylceramide, which leads to really lots of fat and the fatty macrophages of the Gaucher cells, and that gives you the fatty spleen. However, with a very, very small minority of cells, probably much less than 1%, it's possible that the enzyme is blocked a little bit, not too much of the substrate gets increased, just a little bit, but you do have increase of the phosphatidylserine and an increase of the saposin C. So it's not just a big fatty red blood cell. Instead, you have elevated levels of these plus a bit of the enzyme, the glucosylceramidase, and then the macrophage adds the acid. So the acid added from the macrophage activates the saposin C, and instead of having large amounts of the substrate, it drives the reaction forward, and you have an increase in ceramide. Ceramide is the linchpin protein with apoptosis. If ceramide is increased, it can lead to an increase in caspase 8-9, which leads to an increase in caspase 3-6-7, which eventually leads to apoptosis, which is programmed cell death. Now that we know everything we do about Gaucher disease, the very rare occurrence with this very rare disease, and the functionality of saposin C, does BXQ-350 have what it takes to work on cancer? Well, cancer would have to have an acid environment, phosphatidylserine on the membrane, and enzymes available for the saposin C and phosphatidylserine to drive and increase the ceramide. Let's go to the next section and find out. This second section in the second box is titled the backup plan. We'll be covering the biology of normal cells and cancer cells, and we'll also be covering some of the therapies that we use today for cancer. Normal cells tend to work well together, are organized, and are even sacrificial sometimes. Cancer cells do not work well together, are not organized very well, and are the opposite of sacrificial. It's thought that cancer cells hijack the functions of normal cells, and it does them in a very similar way across many cancers. It's possible, if a cell does not die, that it can turn on some ancient programming that makes it want to survive. And you can imagine maybe an ancient time when cells weren't really organized or work well together, that you just need to survive and fight off different cells. Of course, this is just a guess. Normal cells have many, many functions. We'll be covering 4 of them. Normal cells divide or undergo mitosis, so one cell would become 2. Normal cells also grow. They have a number of proteins which encourage growth, so it starts smaller and then becomes bigger. Everything needs energy, and normal cells derive its energy from the mitochondria and uses oxygen, and it helps produce lots of ATP, which are energy units, but no acid is produced. Normal cells also undergo apoptosis. This actually happens billions of times a day. As we know, normal cells have a neutral phospholipid, the phosphatidylcholine, on the outside. As apoptosis occurs, the cell shrinks, and little blooding occurs, and what are called apoptotic bodies form, and they have phosphatidylserine on the outside. Also, within the cell, enzymes are released, and it breaks down everything in the cell. And then the parts after they're broken down are taken up by the phosphatidylserine to be reused in the body. And that leads to the apoptotic cell and lots of apoptotic bodies, and these go to the rest of the body to recycle the parts that are from the cell. Cancer cells hijack these 4 functions. Cancer cells divide at a very high rate, producing lots and lots of cells, and that's been known for decades. Cancer cells also have growth factors due to their cancer, and that may increase the cancer growth rate. Cancers also produce energy. It can still use the mitochondria, but what it normally does, it takes over the cytosol and uses the anaerobic process and produces ATP and acid. Now, this is 18 times less efficient than the normal cell. However, it more than compensates by increasing the rate 100 to 200 times. Cancer cells also hijack the apoptosis process. Now, instead of, as we know, the phosphatidylcholine is on a normal cell, but the apoptosis is hijacked, so only phosphatidylserine is on the outside of most cancer cells. And also they produce tumor microvesicles, which are similar to the apoptotic bodies, and send out little vesicles that have dominant phosphatidylserine on the outside. And these carry DNA and RNA and can actually spread tumors in that way. Also, there's an increase in the enzymes within the cell, but not at the extreme level where it dissolves the entire cell. And some of the enzymes that are released are from the lysosome also. Also, since the apoptosis process is frozen at this point, it's much, much harder for the tumor cells to become apoptotic, to die. Chemotherapy drugs are small organic chemistry-based molecules used to inhibit different parts of cells involved with the mitosis process because cancer cells divide much more quickly than normal cells. As you can see here, chemotherapy blocks DNA and other sites inside the cell. This damages the cell. If there's been a little bit of damage, the cell can fix itself. If enough damage occurs, some unknown internal decision mechanism will decide to push the cell into apoptosis. Unfortunately, chemotherapy doesn't only reach a tumor, which usually accounts for less than 1% of a body, but it also goes into the other 99% of the body, all the normal cells. For example, during a normal 30-minute infusion for chemotherapy, you can actually count out how much goes into the cancer. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Those 18 seconds seem long, but in reality, that only represented 1% of the chemotherapy. On the positive side, those 18 seconds of drugs can reach where no blade and no radiation can reach. On the negative side, the other 29 minutes and 42 seconds of the chemotherapy goes to all the normal cells throughout the body. Chemotherapy agents will attack cancer cells, as you can see here. It's a small molecule, diffuses into the cell, and blocks DNA or other structures. Chemotherapy will also attack normal cells that divide quickly also, as it does here. Small molecule goes in, and the other cells, for example, that divide quickly are the bone marrow, hair, and the gastrointestinal tract. Since chemotherapy drugs are small molecules that diffuse throughout the body into cells, sometimes certain drugs will inhibit unintended proteins. One such example is doxorubicin. Every time it's used, it will actually cause damage to heart muscles. Troponin is a marker used to test for heart attacks. An elevation indicates heart muscle damage. Doxorubicin elevates troponin every time it's used. Personalized or precision medicine drugs are also small molecules that enter cells. They're designed to inhibit growth factors that have been overproduced in cancer cells. Gleevec is one such example. One advantage of these type of drugs is that they are rational design drugs and have less side effects than most small molecule-based drugs. One disadvantage is that each personalized or precision medicine drug works only on one growth factor defect. Cancer has lots of these, and since it takes hundreds of millions of dollars to bring any one single drug to market, that fixed cost needs to be spread to a limited number of patients, oftentimes to populations of 10,000 or even less, leading to very expensive therapies. James Watson was initially supportive of this drug development because it does work. However, when industry, government, and academia started devoting the great majority of their time, effort, and money doing personalized precision medicine, he changed his mind. With his open letter 2 years ago, he was concerned that 1, resistance developed too easily with personalized precision-based medicines. 2, that these drugs do not kill the cancer cells. They're oncostatic, not oncocidal. And 3, he felt that greater attention is needed for the chemo-resistant cancers, especially ones that are p53 negative. He felt and concluded that a biochemical process could be the best way to attack cancer, all cancers. This third box is titled The Best Strategy. Here we'll see how BXQ-350 answers Dr. James Watson's question. It's not just one biochemical process that happens with BXQ-350, but 3 biochemical processes. BXQ-350 is made of 2 large biological molecules. They're normally found in the cell, so they should be well tolerated, unlike small organic chemistry-based drugs. When BXQ-350 travels to normal cells, nothing happens. The outside of normal cells are pH 7, and there's no acid to activate the sapsin C within the BXQ-350. The outside of the normal cells have phosphatidylcholine, so there's no way that the sapsin C can destabilize the membrane to enter the cell. And even if it did, the lysosomal enzymes are not leaking. They're contained within the lysosome. With cancer cells, there is acid on the outside of the cancer. It's a pH of 6. BXQ-350, the sapsin C, will get activated. And then, that's the first biochemical process. The second biochemical process is that since there's phosphatidylserine on the outside, BXQ-350 will destabilize the membrane and fuse and go into the cell. And the third biochemical process, once the BXQ-350, the sapsin C phosphatidylserine entity, is inside, it connects with the enzymes that are leaking out of the lysosome. And if there's enough sapsin C and enough phosphatidylserine and enough enzyme, it will drive the reaction, just like we learned with the very rare circumstance in the very rare disease of Gaucher's. It will increase the ceramide, and that can lead to apoptosis. BXQ-350 can also kill chemo-resistant cancer cell lines. We've seen this picture before. As we discussed before, chemotherapy was a small organic chemistry-based molecule that will damage DNA, which may lead to, if there's enough cell damage, a decision process which we don't know yet, and that can lead to increased ceramide and apoptosis. With some cancers, they mutate, and there is something called p53, which can become mutated and stop this process, the decision going from the DNA to the decision tree from occurring. With BXQ-350, we have a classic military strategy. It flanks. So if you have a blockage like p53, usually you want to either get rid of it or revert it back. And there's lots and lots of research that wants to do that so that you can reuse chemotherapy and get apoptosis. With BXQ-350, it doesn't even matter. You could have normal p53 or the mutated version, and it doesn't matter because it totally skips those steps. It, as we know, it directly goes into the cell, and it increases ceramide and leads to apoptosis. But flanking is not even the best strategy for killing cancer that BXQ-350 does. Cancer cells themselves have a strategy for survival. They produce acid on the outside. The enzymes are released on the inside and actually also ejected on the outside of cells. This acid and enzymes act as an offensive weapon and attack normal cells that are nearby and allow the cancer to spread. And also, if any immune system cells come and try and attack cancer, these neutral immune system cells also have difficulty with the high acid and high enzyme level. So cancers have a strategy of survival, probably one of their best, is to produce an environment that is difficult for normal neutral cells to deal with. So when you think about it, the outside of cancer cell looks very similar to the inside of a lysosome. there's acid, there's phosphatidylserine, and there's enzymes. And that produces the environment that attacks normal neutral cells. So what better drug to give to a cancer than the two components that are in BXQ-350, sapacin C, which is the protein that's normally in lysosomes, and phosphatidylserine. In The Art of War, Sun Tzu said, "The highest form of warfare is to attack the enemy's strategy itself. The next, to attack his alliances. The next, to attack armies. The lowest form of war is to attack cities. Siege warfare is a last resort." BXQ-350 is the best strategy and the highest form of warfare against cancer.